Okay, so we are today going to finish up our review by doing a very quick overview of chapter 4. So after this, we'll be able to move into things that are specifically anatomy stuff. Okay, so today we have different processes that occur that contribute to the metabolism within a cell. So first, we're going to look at the difference between a catabolic and an anabolic reaction, okay, or catabolism and anabolism. In a catabolic reaction, a molecule is going to be broken down into smaller molecules or smaller parts. In an anabolic reaction, smaller molecules are going to be built up into a larger molecule. So in a catabolic reaction, that could be something like a protein being broken down into amino acids, like what happens during digestion. Or an anabolic reaction could be during protein synthesis, an amino acid is forming a peptide bond with another amino acid in the protein chain. Okay, so the thing that we want to note here is energy in the direction of energy. So in an anabolic reaction where we're building something up, that requires energy. So energy needs to be put in so that we can form these bonds. In a catabolic reaction, energy is actually going to be released through the breaking of that bond. Okay, so this says the same thing. Catabolism, large things being broken down, and that releases energy. Anabolism is the opposite, smaller things being put together, and that is going to require energy to do that. So the process behind catabolism is known as hydrolysis. So in order to break something down, a water molecule needs to be put into the system in order for those two things to be split. So here we have a sucrose molecule, and a water molecule is being put in to water molecule is being put in to break this bond right here to split it into the two monosaccharides. All right, so with anabolism, that is known as a dehydration synthesis. So again, this is the opposite. So in order to build something up, a water molecule needs to be removed, okay, hence dehydration. So, an anabolic reaction is going to help with growth. It could involve the formation of glycogen by bonding all these glucose together. It could be the formation of a triglyceride by bonding, like as we see here in this picture, by bonding a fatty acid tail to the glycerol. Okay, so basically the water is coming from the OH group on one molecule and then the H group on a different one. Okay, so they bond together, they're released, and now we have the carbon here now bonded to the oxygen. Okay, so for the next couple slides, I want you to determine whether it is anabolic or catabolic. So here we have a short polymer and a monomer that are being bonded together to form a longer polymer. So is that anabolic or catabolic? Okay, here we have two monosaccharides that are being bonded together to form maltose, so 2-glucose. Here we have sucrose, and water is being inputted, and the enzyme known as sucrase is breaking it down into two monosaccharides, glucose and fructose. So is this anabolic or catabolic? All right, and that brings us to enzymes. So enzymes proteins, globular just means that they're kind of, their shape is kind of rounded. All those amino acids, they come together to form kind of like a round looking glob of protein. And so enzymes are responsible for helping out with metabolic reactions. So they reduce the activation energy required for a reaction to happen. So they really vastly speed up a chemical reaction that occurs within a cell. Right? So they have the ability to actually, by lowering the activation energies, speeding up a reaction by a factor of 1 million or more. So it can make it 1 million times faster, which is crazy to think about. Okay, so here we have an enzyme, okay, that has the site here. That is the active site where it will bind with this protein here and then split it up into two smaller parts. Okay, so it binds here, and then it would split it up. Okay, so the enzyme works on a specific molecule, which is called its substrate. On the enzyme here, so this part is the active site where it will bind to the substrate over here. Okay, so the enzyme plus the substrate is known as the enzyme substrate complex, and the enzyme substrate complex will then perform that reaction. So the enzyme comes out of the reaction and will have either built something or broken something down. Okay, so the important part is that the enzyme does not change. So it is what is called a catalyst. It does not change during the reaction, which then means that it can go and do the same thing again with the same protein, okay, just with a different one. Another example of an enzyme is catalase. Whenever we were talking about peroxisomes, catalase is an important part of peroxisomes because it helps to break down hydrogen peroxide, which is toxic to the inside of your cells. So catalase will break it down into water and oxygen, which we know are good for your cells. Okay, so there are many, many different chemical reactions that are occurring within your cells, which means that you have many enzymes in order to help out with each of these chemical reactions. Okay, and a lot of these reactions are part of a larger thing. Okay, so what is called a metabolic pathway, which is where a bunch of chemical reactions occur in a chain in order to get from one thing to a finished product. Okay, so it helps to really build or break down pretty complicated macromolecules. Okay, so cofactors are chemicals that help enzymes to get there. So proteins need to have a specific shape. So cofactors help them to get the appropriate shape for their active site. Because if the active site is not correctly shaped, then it will not be able to bind with the thing that it needs to bind to, with the substrate that it needs to bind to. Okay, ions or coenzymes are, so vitamins are cofactors that will do this for the enzyme. Okay, now a problem with proteins and enzymes in general is that things like heat, differences in pH, radiation can denature or change the shape of the protein, which will inactivate it because then it won't be able to bind to the substrate appropriately. Okay, so something like cyanide, which you should know is very poisonous to you, can really affect enzymes that contribute to your respiratory functions. All right, so here we have ATP, right? So we have the adenosine, okay, triphosphate. So we have adenine here, we have a ribose sugar, and we have 3 phosphate groups. So we get energy by popping off this phosphate here in a catabolic reaction to form ADP and a spare phosphate. Okay, so in order to build up ATP from ADP, it involves adding that phosphate here. So energy is put in an anabolic reaction called phosphorylation to build the ATP, and then ATP is broken down in a catabolic reaction to release that energy. Okay, so cellular respiration, this is all we're going to talk about it. Okay, so this is not AP Bio where you would learn all the different steps. We're just going to kind of talk a little bit about this. Okay, so cellular respiration is where we are building ATP with the help of glucose. So the energy from the glucose is going into the ADP to build the ATP, so that that energy can then be released at a later time. So the steps are known as glycolysis, which occurs in the absence of oxygen. And then we have the citric acid cycle and the electron transport chain. So glycolysis, again, like I said, can occur without the use of oxygen, but it only forms a few ATP, whereas the citric acid cycle and then the electron transport chain produce many more ATP molecules. All right, so protein synthesis, we're going from DNA to RNA to proteins. So gene on DNA will code for a specific protein. Okay, so it has instructions to make that protein. So the proteins that you have, okay, they manifest your traits from the DNA. Okay, they will be built according to the instructions that are provided on the DNA. Okay, so the central dogma of biology is taking the DNA to form RNA to form proteins. This is transcription right here, where we're taking DNA and copying a specific gene. Okay, so remember, RNA is a single strand, and the RNA polymerase, which is an enzyme, will split open the DNA. The RNA will match, okay, because remember, these nucleotides are complementary to each other. They'll match with one strand of the DNA and copy that gene, and then when it's done, it will come off. So then we have a single strand of RNA. Oops. Okay, so here we go. We have the DNA bubble. The RNA is going to form. The DNA bubble will shift, move down as more of the RNA strand is built. And eventually, the RNA strand will be fully built, and then the DNA will release the RNA, and the DNA will go back to its original shape, that double helix. In translation, we have the RNA here, which can be split up into 3-letter words, okay, which code for a specific amino acid. Okay, so 3 DNA nucleotides will build 3 RNA nucleotides, okay, because they're 1-to-1, but then 3 RNA nucleotides will code for 1 amino acid, right? Because remember, we only have 4 different nucleotides, but we have 20 different amino acids. So it's important that, the alphabet here, which builds 3-letter words, okay, will code for different amino acids. Okay, so translation is the process that we're talking about here, going from RNA to protein. It occurs at a ribosome, and basically the RNA is just pulled through the ribosome, and every 3-letter codon on the RNA will code for a specific amino acid. So these tRNA will come in and translate, okay, by matching up with the codon, and then they provide the specific amino acid here. So they're translating between the nucleotide and the protein. the nucleic acid in the protein. All right. Okay, so eventually, again, the RNA strand will get pulled through the ribosome, and then so more and more of these tRNA molecules will come in and provide the next amino acid in the chain. Okay, so peptide bonds occur between amino acids. Again, look, OH and H. This is an anabolic reaction. This is dehydration synthesis, because these are going to get pulled off to release a water molecule. And then we can go to the next amino acid in the chain. Okay, so peptide bonds, these are anabolic reactions. Okay, now the protein structures. So we start off with the primary structure, which is just a sequence. And then the amino acids will interact, forming hydrogen bonds and then even covalent bonds as you get to the higher structures. Now, a very important part of protein synthesis is that mutations can happen. Now, there are a lot of safeguards that help to reduce mutations, but they can still happen. So, a mutation can be something as simple as just substituting a different nucleotide here, which could mean that the amino acid is changed. Okay, in this case it is. But a nucleotide could be removed or added, which instead of just affecting one amino acid, will affect every amino acid after that addition or subtraction of a nucleotide. Okay, so you see here with the substitution, not only affected one amino acid, but here it is affecting all three of these and possibly more after this, depending on how large the protein is, okay, or how close we are to the end. Okay, and that is the end of chapter 4. This is a very, very quick run through. So if you have any questions, please let me know. But we are going to do a little bit of practice with these catabolic and anabolic reactions tomorrow. All right, so just keep that in mind. And with that being said, I hope that you have a nice day.